Excellent. Looks like everybody's starting to come in. That's
Okay.
great. Get started here in just a minute. Okay, looks like things are slowing down, so we're going to go ahead and get started. Just want to welcome our MPN research advocates. We are thrilled to have you here for our second session. We also want to thank you for really your interest in helping shape MPN research. For any new participants, I do want to introduce myself. My name is Sarah Douglas, and I am the Associate Director of Patient Engagement here at MPN Research Foundation. We really want you all to always feel comfortable reaching out to us with any questions you have along the way of the MPN Pathways program. So please, at any time, reach out to me at patientengagement@mpnrf.org. For tonight's presentation, we will have Dr. Weir answer a few of the group's questions live. So please place those questions in the Q&A feature during the presentation, and they will be addressed at the end. So anytime a question comes up, just put that in there so you don't forget it, and we'll get those at the end. And then as a reminder, tonight's presentation is being recorded, so that link will be provided to registrants after the webinar. And as mentioned, this is the second session of the MPN Pathways series. So just as what's coming up, so the third session is titled MPN Clinical Trials 101. This is going to occur on January 23rd of 2025 from, again, 5 to 6 p.m. Central. MPN Research Foundation Senior Manager of Research Programs, Christina Prasad, will be discussing the significance of clinical trials in developing new therapies and improving outcomes for MPN patients. A registration link will be sent out following this webinar, so we'll make sure that you guys have the opportunity to register for that right after this. And then next year, just to kind of a prep, we will be learning about the FDA. We'll also be learning about patient and caregiver voices and research, and then taking a deeper dive into some of those clinical trials and research protocols. But it is now my pleasure to introduce our speaker for the evening. We have Dr. Scott Weir. So Dr. Weir is a member of the MPN Research Foundation Scientific Advisory Board, and he holds many important titles at the University of Kansas Cancer Center. Dr. Weir is the director for the Institute of Advancing Medical Innovation. He is the Associate Director for Translational Research. He is a professor in the Department of Cancer Biology and Medical Oncology, and he is the Kelly Family Foundation IAMI Professor in Cancer Drug Discovery and Development. Also to be noted, Dr. Weir has a passion for working with cancer patient research advocates and really ensuring that their voices are represented as therapies really move from the lab to the bedside. But with that, Dr. Weir, thank you for being here. I will hand things over to you and just let me know when you're ready to advance slides. Again, thanks for being here with these patients and caregivers.
Absolutely. Yeah. Thank you, Sarah. Yeah, if you would move to the next slide. Yeah. Hello to all of you, and thank you for joining here. I guess it's this evening, my time. And certainly thank you, Sarah, and the MPN Research Foundation for the opportunity to talk with you here for a few minutes this evening about the journey of bringing new therapies to patients. And certainly what I'd like to do is at least share some of my experiences in being in this business for now almost 39 years. And certainly what we're trying to do really to improve the treatment outcomes for all cancer patients. So next slide, Sarah. So what we'd like to cover with you here today is one, to talk a little bit about drug development, and hopefully you'll get an understanding of the journey of bringing new therapies to patients. Learn a bit about the different types of cancer therapeutic agents, and a concept that really is central to what we as drug hunters think about, which is referred to as a drug target. I'll describe the clinical trial phases in which potential or promising cancer therapeutic agents journey as we go through the clinical research process. and then also describe the FDA and regulatory agencies, honestly, throughout the world, but the review and approval process that companies and biotech companies work with regulatory agencies to get clearance to commercially market their drug products. Next slide, Sarah. So this business of drug discovery and development is an expensive and incredibly high risk endeavor. And traditionally, the process, a lot of folks really like to describe it really as a funnel that's wide at the top, and hopefully you can see in this slide, it narrows as projects advance through the drug discovery and development process. It starts with drug discovery, where if it's a what we call small molecule drug, and I'll describe that here shortly, but we start with hundreds to thousands to perhaps even tens of thousands of chemicals or compounds that aren't quite drugs yet, and the majority of them will not become drugs. And then we go through a series of discriminating tests, both in the test tube and certainly in animals, and as well as in humans, to be able to determine which of these compounds have the greatest potential of becoming an approved drug, and certainly those that don't hold promise that we can eliminate as quickly as we as quickly as we can, to really focus on the most promising compounds moving forward. So this process for drug discovery and development, including the regulatory review and approval process, and then commercial launch, typically takes anywhere between 10 and 15 years, sometimes 17 years. Some folks estimate that the fully burdened cost to bring a new drug to market today is in excess of $2 billion US. And again, with a journey that requires 10 to 15 years, sometimes there's not much patent life left that the company is able to recoup its substantial investment. So again, in the context of chemicals or small molecule drugs, the average is only 1 out of every 10,000 compounds makes it through this process to market. Next slide, Sarah. So in cancer, there are different types of therapeutic agents, and we've shown here chemical structures of 3 of these types. And in the lower left-hand corner is a small molecule drug, and you can see the relative size of this agent here. In the middle, in the kind of, I guess it's pink or purple, is what's called a short peptide. It's a little bit larger than the drug on the left-hand side. But again, it's called you could say a medium size or short peptide drug. And this particular agent, cyclosporin, was originally developed and approved to be able to give to patients who are receiving organ transplants. And then on the right-hand side is perhaps one of the most famous cancer immunotherapy agents, called pembrolizumab. And it's what's called a biologic. And you can see there that this drug is actually a monoclonal antibody, and it's huge compared to the others. Now, certainly the academic biotech and large pharmaceutical research community were discovering and developing each of these types of agents. And just to kind of compare and contrast, the small molecule drugs, again, illustrated by the funnel on the previous slide, they are chemically synthesized and manufactured. Typically, these types of drugs are stable. They can be stored at room temperature. And so, for example, if you've got a prescription in your bathroom and it's a capsule or a tablet, obviously you know that you're able to store that drug product, at room temperature. Often small molecule drugs are able to be given orally. And once they're absorbed into the bloodstream, they'll distribute throughout the body. The cost to develop a small molecule drug is relatively low compared to what I'll describe on the right-hand side of this slide. But there's an incredibly high failure rate. And as I mentioned, 1 in 10,000 chemicals actually becomes a small molecule drug. And certainly for the MPN community, Jakafi is an example of a small molecule drug. Now back to pembrolizumab or the biologics. These types of drugs are typically derived from living organisms. They're manufactured by either yeast or bacteria or mammalian cells. And they would include examples like vaccines, monoclonal antibodies, immunomodulating agents, and growth factors. Now, these large molecule biologics are very often, if not routinely, unstable at room temperature. And they're given by injection either directly into the bloodstream. And because they're so large, they will remain in the bloodstream and the lymphatic system. And their ability to distribute outside of the circulation is really limited by their size. Now, the development costs are quite high, particularly around manufacturing these types of drugs. But there's a relatively low failure rate. The number of candidates is nowhere near, 10,000 to be able to get one pembrolizumab drug. So Sarah, the next slides, please. So what I'd like to do on the next few slides is to describe the drug discovery process. And it starts with, on the left-hand side of this slide, where we make a biology discovery. And that could be where a biologist working in the lab has made some discovery that is associated with a normal cell kind of going haywire or transforming from a normal healthy cell to a cancer cell. And that biological observation or event is something that we as drug hunters then think about in terms of, is this something we can turn off or perhaps turn something else on to either kill the cancer cell or prevent it from transforming from a healthy cell to a cancer cell. As described previously, we then have chemists who will do exploratory chemical reactions to potentially synthesize drugs that can either flip that light switch on or turn something off. And then we'll, if we can identify probe or tool compounds that have these abilities, then we like to try and improve the properties of these experimental agents to then enhance the success, which is ultimately bringing a drug to market. We'll take these compounds into typically test tube experiments where we're evaluating their activity in killing or modulating cancer cells. And from there, from those experiments, we'll select the most promising compounds that we would then take into animal studies. And if we can work through this process from the left to the right and actually establish that a potential therapeutic agent has activity in a validated animal model of cancer, at that point, then we'll consider that compound graduating into formal drug development, where, and I'll describe what's entailed there shortly. Sarah, the next slide, please. So when we, in discovering and developing anti-cancer agents, we typically think about what's called the hallmarks of cancer. And there were some investigators that in 2000, 2010, and then here I've given the reference below. In 2022, these folks really published landmark papers that describe what's called the hallmarks of cancer. And these are the mechanisms, the processes, and the traits that cancer cells develop in order to grow, in order to survive treatment, and then of course put the lives of our patients at risk. And so when one of our cancer biologists makes one of these laboratory discoveries, those discoveries are really put in the context of this diagram here with the circle going around and these hallmarks of cancer. So again, these are the things that we're really thinking about in terms of, can I shut something down or turn something else on that will make it more difficult for a cancer cell to survive? The next slide, Sarah. So I mentioned earlier that we think about drug targets. And again, as I mentioned, that a drug target is a biological process that goes on in a cell that in the context of cancer may be associated with a normal cell transforming into a cancer cell. And as we're discovering drugs to attack that target, we're really thinking in most cases about a therapeutic agent that can directly attack the cancer cell. And that's shown in the center here with this arrow being shot by the bow that's going directly to the bullseye. And then on the left-hand side, there are also therapeutic agents, cancer therapeutic agents, that are able to block the communication between cancer cells and other cells. And we certainly know through the hallmarks of cancer and the research that's been conducted over the last few decades that there are non-cancer cells that communicate with and send signals to cancer cells that are able to assist those cancer cells in surviving and growing. And then in the lower right here, there are also therapeutic strategies where we're attacking the local environment, let's say around a cancer cell or a tumor. And by attacking that local environment that supports that tumor's growth and survival, we're able to kill those cancer cells. So again, this is the concept of a drug target. Next slide, Sarah. So, and what makes a good drug target? Well, when we look at these biology discoveries, what we're really looking for is a discovery of a process that is specific to a cancer cell. If there's a process that's discovered that is common not only to cancer cells, but normal healthy cells, and we shut that process down, we run the risk of having off-target or toxicity to normal healthy cells, which certainly is not the goal. Ideally, the target should be found in a cancer cell. And then also the target should be found in a location that can actually be reached by a drug that is administered by routes of administration. And whether that's orally, intravenously, et cetera. So the next slide, Sarah. So in terms of an example of an anti-cancer agent directly attacking a cancer cell, this figure here shows a tumor cell in the center of this picture. And this is actually a malignant multiple myeloma plasma cell. And for those of us that remember COVID on the news back a few years ago, and I can't believe it was 4 years ago, but you may remember the pictures of the virus, and the virus had many spikes sticking out of it. And it's similar with cancer cells. And on a multiple myeloma cancer cell, it has some spikes sticking out of it. And in the majority of these patients and in the majority of these cancer cells, the spike is referred to as BCMA, or B cell maturation antigen. And with one of the immunotherapy strategies that we use today and is continuing to grow is called CAR T cell therapy, where we collect T cells, immune cells from patients, we reprogram them, and in the case here, to be able to go out and find this spike, this BCMA, and then bind to it, and in doing so, kill the cancer cell. Now, in the case of CAR T cell therapy, this CAR T cell will bind directly to the tumor cell and kill it, but also it's recruiting other immune cells, other non-engineered T cells that come in and assist the CAR T cell in killing the cancer cell. So Sarah, the next slide, please. So in terms of blocking the communication between other cells and cancer cells, on the left-hand side here, what Sarah and I have shown for you is a cell that basically is spitting out little chemicals, and they're illustrated here by the green balls. And it sends these cells out of the cell into the local environment. And then these signaling molecules, these proteins, then are able to travel to the target cancer cell, and they're able to engage with this red thing here, which is the target or the receptor that's at the surface of the cell. And so when that green ball travels across and then fits inside that pocket there of the receptor, it's able to then promote the signaling within the cell that facilitates the cell growing, invading healthy tissue, migrating from the tumor to elsewhere, and spreading throughout the body. And so what we do as drug hunters is to be able to interrupt this communication. We're discovering and developing these little yellow balls that can actually be administered and then travel to that target and actually sit on that target receptor and block the ability of the little green balls to be able to engage with that target and promote survival of that cancer cell. So this is, again, an illustration of how, as drug hunters, we're trying to block the communication between a cancer cell and another cell that assists in its survival. The next slide, Sarah. And then the third area really is attacking the tumor microenvironment. And we certainly know today that a tumor is not comprised solely of cancer cells. Tumors survive and grow because they're able to develop a blood supply. They're able to develop an infrastructure that physically supports the tumor. And then there are a number of different types of immune cells that also coexist with the tumor cells that, again, promote that tumor's growth and survival. And so certainly many of us are working through different mechanisms to be able to attack what's called this tumor microenvironment. And certainly there are drugs on the market that will shrink blood vessels to hopefully decrease the blood supply to a tumor, and then also attacking these immune cells that support the tumor's survival and growth. The next slide, Sarah. So it's a bit of an abstract concept, but if you would maybe for just a minute think about and Sarah, maybe if we could just hold off there for just a second. Yeah, thank you. So when we have a target, and if you can again envision that little red target receptor 2 slides ago, when we make this discovery that there's a target receptor that a cancer cell relies upon to be able to survive and grow, we have these folks called structural biologists that are actually able to conduct experiments that characterize the three-dimensional structure of that target. And when we're able to do that, then what our chemists do is they design the structures of molecules that are actually able to go in and fit within that receptor or within the binding pocket of that target. And so if you would, Sarah, go ahead and flip the movie on. And so this is an example there. You'll see a small molecule traveling to the receptor. And once it finds the binding pocket, it's going to actually insert itself in there. And then that, let's say, blocks the communication between two cells, will cause the death of immune cells that are supporting the tumor microenvironment, et cetera. So as drug hunters, this is what we think about. We characterize the three-dimensional structure of a target, and then we design molecules that will actually bind or engage directly with that target. And so we like to use as an analogy, let's say you've got your iPhone and you need to charge your iPhone, and you plug the charger into the bottom of your iPhone. It's a similar concept, or another analogy would be a key fitting into the lock. And so if the lock is the drug target, you design and you shape the key to be able to sit directly into that lock. So Sarah, the next slide. And so this is just a different illustration. On the left-hand side of this slide is the target and the binding pocket for that target. And then this is the chemical structure, the three-dimensional structure of the molecule that was designed to fit into that binding pocket. On the left-hand side, and I apologize, the structures might be a little small for you to see, but it's an example of the very first approved anti-cancer agent that actually controls cancer. And the drug here is called imatinib, and or Gleevec for some of you that may know the trade name. And what the team at Novartis did was, as they structurally characterized that receptor, that target, they then synthesized different variations of imatinib and then tested their abilities to be able to bind either more strongly or perhaps less strongly within that binding pocket. And by making all of those derivatives, and again, I don't believe they synthesized 10,000 of these small molecule compounds, they ultimately tested and identified that imatinib did the very best job in engaging with that drug target. And so this is the chemistry approach that we take in discovering new agents. So the next slide, Sarah. So let's say once we've done some of this initial foundational biology and chemistry work, and we've got some compounds like on the right-hand side of the previous slide, then we really ask ourselves and start with testing in cancer cells. but we ask ourselves and we ask of the compounds, is the compound active? If it's active, is it potent? And is it stable? Is it stable in the bloodstream? Is it stable in the body? And then also do experiments to try and figure out, okay, how exactly is this compound working to kill cancer cells? And we use this information to then select the most promising compounds that would go into animal studies. And in animal studies, we're asking basically the same questions. And we're asking, does drug get to the tumor? If so, so let's say if you give it by injection or give it orally, does it distribute in the body and actually able to reach the tumor? And if drug does reach the tumor, does the potential drug actually engage with the target at the site of action? And then with that drug target engagement, is there a therapeutic response in an animal model? And for example, do tumors get smaller? Okay. So the next slide, Sarah. And so provided we've been able to establish this, at that point, then the very best compound is promoted to what we call a development candidate. And at that point, we have to conduct studies required by the regulatory agencies throughout the world that will enable us to be able to advance that drug candidate into humans for the very first time. And so we've got to conduct experiments to characterize the toxicity of that compound in animals. We've got to characterize the potential untoward or off-target effects of that development candidate in animals. For example, does it alter blood pressure or respiration, or does it cause drowsiness? We have to manufacture that drug in a high quality environment that would be suitable for administration to humans. We've got to formulate it into a drug product, whether that's an injectable or a tablet or a capsule that can be given to humans, and then manufacture sufficient quantities to support clinical trials. We will also then design the very first in human phase 1 trial. And again, we'll talk about that here shortly. And then also lay out or define for the regulatory agencies what our plan is to be able to investigate the safety and efficacy of this potential cancer therapeutic in patients. It's quite a similar process, whether you're seeking clearance from the US Food and Drug Administration or the European Medicines Agency in Europe or the Ministry of Health in Japan. But all of these data are compiled into what's called either an investigational new drug application in the case of the US, or a clinical trial agreement application for those other regulatory agencies. Now, provided the agencies agree that the scientific evidence warrants evaluating this agent in humans for the very first time, we then move into clinical development. And so the next slide, Sarah. And I'm sure many of you have heard these terms. We start with what are called phase 1 studies, where these are studies that are really designed to characterize the human safety of a development candidate, and really to not only characterize the safety and the tolerability of this agent, but to also define through that process what's the greatest or the maximum safe dose that can be given to humans. Now, once that's established in phase 1, then we'll move to studies where we will evaluate a range of doses in a larger population of patients to be able to then to begin to characterize what are active doses, what are active anti-cancer doses, and what are the safety profiles of the agents as a function of dose. And then from there, then the optimal dose is selected, which is then evaluated in phase 3 safety and efficacy trials. And these trials could typically include anywhere from 250 to 2,500 patients, depending on the indication. Provided that the drug demonstrates safety and efficacy, then the company will prepare a new drug application and submit that to the regulatory agencies, and typically throughout the world, to seek approval to then market and commercialize the product. I would say most often, and certainly this is the case in cancer, beyond regulatory approval, there are phase 4 studies that are conducted to further characterize the safety, but also answer other questions that emerge through the clinical development of the particular agent. And certainly in the case of cancer, drugs will often be approved for one indication, but there may be therapeutic applications in other cancer types and other types of cancer patients as well. Okay, next slide, Sarah. So as we're developing, as we're advancing a candidate into clinical trials, we really ask ourselves, is this potential therapeutic agent, should it be given as a single agent or in combination with standard of care? And that really drives how you design the early phase clinical trials. So in the case of a single agent, you're really looking to achieve improved treatment outcomes over the current standard of care. But often what we're doing is we're adding the experimental therapeutic agent to standard of care and seeing if we can improve treatment outcomes above and beyond the outcomes associated with standard of care. And then as well, we're asking ourselves, should this potential cancer treatment be given in a neoadjuvant setting, which would be before standard of care treatment? For example, if there's a tumor and the patient is scheduled for a curative resection of that tumor, would you administer the investigational agent to shrink the tumor or kill, any cancer cells that are beyond the tumor? Or should the agent be given as an adjuvant, either in combination or after standard of care? So the next slide, Sarah. And then the clinical trials, some of you may have heard of the term or the terms umbrella trials or basket trials. And certainly early in development, an umbrella trial is where one will enroll one type of cancer patient, but be able to enroll patients with different gene mutations, and then be able to, as the patient is enrolled in the trial, determine what the specific gene mutation is that's driving that patient's cancer. And that mutation will then drive which specific treatment that patient will receive. And so as an example here, 3 different treatments would be prescribed to patients corresponding to their specific gene mutation. On the right-hand side, we certainly know that targets, cancer drug targets, are often found in multiple types of cancers. And in a basket trial, we will enroll different types of cancer patients that share a common gene mutation. And in that case, they will receive the one treatment that is targeting that gene mutation. So for example, in a basket trial, or let's say you have a solid tumor, we may enroll a patient who has head and neck cancer, who has that mutation. We may enroll a patient who has melanoma with that same mutation, et cetera. And then those patients will receive the one treatment. Okay. Next slide, please. So just to kind of talk about the outcomes, I mentioned drug discovery development is an incredibly high failure rate endeavor. And as we establish that a drug has acceptable safety in animals, and we advance that drug into humans for the very first time, who are participating in phase 1 trials, about 70% of the drugs entering phase 1 will advance to phase 2. And again, in phase 2, we're looking for an efficacy signal. We're looking at where the drug, what the drug does to the body and what the body does to the drug. And about 1/3 of these agents that enter phase 2 will advance to pivotal phase 3 trials that really are designed and conducted to support regulatory approval. And so in phase 3, we're asking, is it safe? Is it effective? Is the risk-benefit ratio for this particular agent in this particular disease, is that acceptable? And then also we're characterizing drug-drug interactions and drug-disease interactions. For example, do you have to modify the dose for someone that might have kidney function impairment? Now, in phase 3, about 1 out of every 4 drugs will fail. And that was certainly the case back in the mid-'80s when I started in this business. And unfortunately, it's still the case today. And this really is the challenge. So for agents moving from the animal phases of investigation into phase 1, only 7 out of every 100 of these agents will actually gain regulatory approval. Next slide, Sarah. So again, just kind of illustrating, these are a little bit different data, but again, similar, where I just mentioned the odds of advancing from phase 1 to regulatory approval are about 10%. Next slide, Sarah. So really just to close, and love to get your feedback, your questions and suggestions. As Sarah mentioned, what's critically important to our program, and not only our program at the University of Kansas Cancer Center, but certainly to pharmaceutical companies, biotechnology companies, is that patients have a voice. And certainly in our program at Kansas, we engage patients, and patients really drive our research agenda. They engage with our researchers to ensure that our researchers are conducting research that's focused on the critical questions that our patients have and the challenges that they face. And so at our program in Kansas, for example, we have over 130 formally trained patient research advocates that really guide us to focus on conducting research on the most important questions. So with that, we'd be happy to answer any questions. Oh, and I'll just close with this slide, which is, again, just in summary. As you go through this process, as we're answering the critical questions for a new therapeutic, as we move through the drug discovery and development process, we're generating more knowledge, we're answering those questions, and better understanding not only how this drug works, but is it safe, is it effective, and how might it best be used to be able to achieve the kind of treatment outcomes we want for our cancer patients. I mentioned that it takes 10 to 15 years through this process, and again, in excess of $2 billion. And so hopefully this talk is helpful to you in understanding the process that we all work through to bring new cancer treatments to our patients. Thank you.
Thank you, Scott. It looks like we do have some questions in our Q&A. I know there's a few in our webinar chat, so if we don't get to all of those, we'll be sure to email you the answers. But it looks like we've got 4 questions. I can read them off, Scott, but I don't know if you can see them.
No, I can.
Okay, I'm going to read them off.
Okay,
though.
thank you.
So our first question from Bruce is, with such a high cost of bringing a drug to the market, how does a relatively small cohort of people, so say our MPN community, or even smaller with our subsets of MF, ET, or PB, warrant the attention needed of pharmaceutical companies that need an economic return? It's kind of a tough question. It says, as an example, the monoclonal antibody work being done in South Australia is a potential cure for people with the CALR mutation, but the investment needed on getting the production may outweigh any potential return for pharmaceutical companies.
Yeah, it's a fantastic question. Thank you. It historically has been a challenge, but I think there's help on the way, so to speak. Certainly, when I joined the pharmaceutical industry back in the mid '80s, and as an example, we were developing a drug for breast cancer, and back at that time, we thought there was one type of breast cancer, and so we conducted a pivotal clinical trial and showed that it only worked in 18% of the patients. And back then, not wisely, we killed the program. Well, in the case of breast cancer, we know there are many, many, many types of breast cancer today. And certainly today, what we would do is we would develop a test that will identify those 18% of patients who would benefit from that drug, and then align that test to be able to then guide oncologists to be able to select the right drug for that patient. There are the regulatory agencies certainly are incentivizing the research community to invest in projects that are designed to treat rare diseases. And there's a number of examples in a number of different therapeutic areas. And certainly there are financial incentives that the regulatory agencies provide, whether it's extending patent coverage, whether it's providing vouchers, or working with regulatory agencies to accelerate the review and approval process. These particular tools are now of access to biotech and pharma companies to be able to move therapeutics forward.
Excellent. Thank you, Scott.
They,
Yeah, Rick.
Sarah, yes, there's a lot of questions, many of them relate going back and forth regarding the disease and asking some specific questions. And I think we should try to get to the questions about drug development, drug discovery and development maybe first, and many of the others which don't relate directly, I think, to the presentation, we can either try to get to toward the end or answer them separately.
Absolutely.
So I would just, I saw a question, now I just lost it, something happened, but give me a second.
While you're looking for that,
Yep.
there were a few questions related to AI,
Yeah,
Scott. I don't
that's
know.
the one I was looking for.
Oh,
Yeah, thank
we're
you.
on the same page.
Yep.
But could you discuss really how AI is assisting or assisting in the future of the drug development process?
Absolutely, absolutely, and we use and certainly the industry uses artificial intelligence in a number of ways to improve and accelerate the drug discovery and development process. And maybe just a couple of examples. if you recall from one of the slides, and I described how we characterize the three-dimensional structure of the drug target, and then with that, being able to define the shape of that binding pocket. We use artificial intelligence to artificially synthesize the compounds that would fit best into that binding pocket, or alternatively, use artificial intelligence to design the specific shape of that key that would unlock the lock. As well, when it comes to, once we've identified a drug that looks promising in animals and early phase clinical trials, artificial intelligence is being used to gain efficiencies in how that drug in powder form is manufactured at lower cost and more quickly, and also using artificial intelligence to help guide how to specifically formulate that drug in powder form into, the appropriate injectable vial or tablet or capsule. And then as well, as we folks are using it where we integrate information on the pharmacology of a drug. In other words, what does the drug do to the body? With disease information, to be able to then look for opportunities to what we call repurpose drugs for new indications. In other words, identify a drug that might, although it may be prescribed for allergies, might have anti-cancer activity in, say, pancreatic cancer or something. So certainly along the continuum, absolutely, we're using artificial intelligence. Great question.
Yeah, thank you, Scott.
Say, Scott, there was another question about whether MPNs are eligible for the FDA's orphan drug pathway.
Yeah, thank you, Rick. So the definition of an orphan drug is less than 200,000 Americans with the disease. And so for a company that is developing a drug for a rare disease, you would submit an application to the Food and Drug Administration seeking what's called orphan drug designation. And once that is granted, and then you complete development and submit your new drug application for approval. In the US, the government will grant you 7 additional years of patent life, of exclusivity. In Europe, that number is 10 years. And so certainly that, again, is an incentive to encourage folks to develop drugs for rare diseases, and certainly including the MPNs.
Excellent. Any other top ones, Rick, or did we lose Rick? Oh, there he is.
I'm here. I'm trying to. A lot of the questions really were more medically related sort of questions, but maybe just there's a bit of a theme going through some of the questions regarding sometimes the difficulty when a potential drug or drug target that's being approached by the academic community, and it's not been, for a variety of reasons, perhaps picked up or embraced by the pharmaceutical community and to for its advancement, right? Or it just doesn't get the recognition that perhaps it would. I wonder if you have just some thoughts, again, especially given the world of repurposing drugs or finding compounds that may show preclinical activity but just not getting a lot of attention from sponsors, drug sponsors of drugs.
Yeah, it's another great question. So maybe there might be, Rick, two questions there.
Yep.
The first, in terms of one of the opportunities, and Rick, this is where you and I first met, is certainly we have a great deal of interest in finding new uses for approved drugs. And so, for example, as we characterize a drug target and we begin to screen tens, maybe hundreds of thousands of chemicals to see if they have activity against that target, right alongside those experiments, we're also screening. We have a library of FDA approved drugs, and we're screening those as well. And to be able to look for opportunities where maybe an FDA approved drug that no one had ever studied, let's say, has anti-cancer activity. And if we can identify those opportunities, at least with some of the drug development processes, you don't have to reinvent the wheel. You can often move more quickly into an early phase clinical trial and see if that FDA approved drug has activity. I mentioned the basket trial, where you have one drug and one common gene mutation, but multiple cancer types. And I gave the example of let's say, head and neck cancer and melanoma. We actually conducted a clinical trial at our cancer center where we evaluated atorvastatin, one of the drugs that lowers cholesterol, that has activity against a specific gene mutation, and we actually studied it in that environment. And in being able to do that, then the academic community can potentially generate enough clinical evidence that would then support off-label prescribing of that agent, for other indications, or build the business case for doing the formal trials and get approval, FDA approval for that indication. The other question, I guess, Rick, about what if there is an academic discovery that is promising, but the investigator can't just get that interest from the companies, and certainly where I work, it's always a challenge. But I think the academics can help themselves by ensuring that they're conducting quality research and they're answering the critical questions that the private sector, that the companies are going to be asking of them in evaluating whether they could take that discovery on and bring a new drug to market. The other is organizations like the MPN Research Foundation and Leukemia and Lymphoma Society, I think, are playing absolutely critical roles in funding this type of research to be able to then generate more compelling evidence that then would encourage the private sector to get interested and potentially partner to bring a therapy forward. So certainly that's been, Rick, that's been my experience.
Yep. No, I agree.
Yeah,
one other one, I guess. Oh, sorry,
well,
go ahead.
I think we're close to time. So I think,
Okay.
Scott, what we'll do is we'll collect these questions, and then kind of similar to what we did last time, we'll send the answers out to the entire group once we've correlated some responses. But Scott, thank you again for being here. You are such a wealth of knowledge, and this was super helpful to kind of understand the huge process behind drug development. So we greatly appreciate it. I have
Absolutely.
a few more slides. Just again, we want to take a moment to recognize and thank our MPN Pathways sponsors and our global collaborator, the MPN Advocates Network, for their support of this program. The evaluation, so this evaluation recording of the session will be sent to you in the post-event email. So be sure to complete this evaluation for this session to earn credit towards your MPN Pathways certification. And then as a reminder, you will be sent the link to register for the next live session in the post-event email. All participants must register for each individual webinar to gain access to the live events, but know that all of these are recorded, so we'll be able to send these out. And then last but not least, I also wanted to make this group aware of a webinar that's happening on December 3rd from 5 to 6 p.m. Central. It's titled Shaping the Future of MPN Research. So during this session, we will take a deep dive into our achievements in advancing MPN research. We will share with you and kind of learn the ongoing efforts to slow disease progression and really how you can be a part of our future vision for breakthroughs in 2025 and beyond. The key speaker will be Dr. Marina Kremenskaya, and I will also include this link in the post-event email, but please let us know if you have any questions about that. Again, thank you, Scott, so much for being here, and thank you all for spending your Tuesday evening with us. We really appreciate it.
Absolutely. Thank you.
All right. Thanks, everyone. Have a good evening.
Bye-bye.
Bye-bye.